Just before we get started, my name is Leia. Thanks for joining our CellCAN webinar. I'm going to give you a couple of details about the projects that CellCAN is working on and who we are before I hand it off to Sam Wadsworth to tell you all about bioprinting at the nexus of medical device, biologic, and cell therapy regulation. So first about CellCAN. We were incorporated as a not-for-profit in 2014 with funding from the Networks of Centers of Excellence. Our focus is honed on the cell and gene therapy manufacturing niche, and our vision is that the cell and gene therapy manufacturing in Canada must operate under a common seal of quality to increase capacity. And our mission is to improve the quality, safety, and feasibility of cell and gene therapy in Canada through optimal manufacturing practices. Our efforts fall within our 4 business pillars. So first is building a strong Canadian network. So this centers around sharing best practices and creating common quality standards to increase collaboration and break down silos between manufacturing centers across the country. Second is advancing regulatory standards. CellCAN works closely with Health Canada and their affiliates on efforts such as consulting on regulatory modernization that's taking place in Canada right now. We want to increase legitimate authorized clinical trials and therapies in Canada while limiting unproven fee-for-service therapies that are becoming increasingly present in our ecosystem. Third, we want to generate effective outreach. We'll do this through our unique training programs, which I'll tell you a bit more about later, and also partnerships and programs like this, our webinar series. We want to get the message out to as broad an audience as possible. And through all of these, we want to position Canada as a world leader in cell and gene therapy production, with a focus, again, on quality, safety, and efficacy. So last year, we hosted a very successful GMP manufacturing workshop. This was hands-on. It was in Montreal at the CETC. More editions will follow. Our plans have been a little bit changed with all of the coronavirus things that have been happening recently. But if you head over to our website, you can sign up to be on the wait list. We'll be sure to contact you with more things, more information as it comes up. Under advancing regulatory standards, we have a cell and gene therapy stakeholders group. We meet twice per year with Health Canada to discuss regulatory issues and also get clarification on the most pressing matters. So if you have any regulatory related concerns, if you need clarification on regulations and guidelines, or if you want to learn how you can contribute to consultations and other regulatory matters, be sure to get in touch with us. We can put you in touch and help you with that. In terms of our upcoming events and forums, we did host a strategic forum in 2019. It was very successful. We're bringing it back in 2021. It's going to be in Ottawa. If you have any new developments you want to share or if you want to learn more, please get in touch. And we are still going to try to head to ISCT. Right now it's still on, just possibly postponed, so we'd be happy to see you there. We're going to be hosting a Canadian pavilion with other Canadian groups. So that's all I have about CellCAN. With that, I would love to hand it over to Sam Wadsworth, the CSO of Aspect Biosystems. Just before I do that, if you have any questions, feel free to type them in. On the right-hand side of your screen, there should be a chat. You can either ask your questions in the chat there anytime during the webinar. And after the webinar, when we go into our Q&A period, if you want to share your voice, I know it can be a little isolating at this time with everyone in a little bit of a lockdown mode. You can unmute yourself by clicking the little microphone either next to your name or at the top of your screen. But please only unmute yourself during the question period. If you just want to type your question in, I will read it out for you. And with that, I will let Sam get started.
Okay, well, thank you very much, Leah. I just want to confirm that you can all hear me. So, Leah, could you just confirm that my voice is coming through okay?
Yep, you're coming through loud and clear.
Fantastic, thank you. Well, thanks everybody for joining today. My name's Sam Wadsworth. I'm CSO of Aspect Biosystems. Unfortunately, I'm not going to talk to you today at all about coronavirus. So this is going to be something different from, I guess, the latest news that you've been hearing about. I'm also not going to talk very much about cells and cell therapy. I'll mention it, but really the focus of today's talk is going to be around the technology of bioprinting and how 3D printing and specifically bioprinting can contribute towards the development of implantable therapeutic devices and tissues. And specifically, I'll talk about how these tissues and devices can be optimized as far as their function and how they may be regulated in the future. My disclosure is obviously I'm an employee of Aspect Biosystems. So here at Aspect, we're based in Vancouver in Canada, and our hypothesis as a company is that we think there is going to be a paradigm shift in the way we develop therapeutics, both drugs and medical devices. And what we've seen over the last few decades is a shift in the way that therapeutics are really developed. And so drugs have really progressed from purely small molecule inhibitors through to more biological type therapeutics. So we saw the advent of biologics, antibodies, and then more recently we've seen the development of cell therapies, such as CAR T therapy. And so therapies are becoming more biological, but also to some extent more personalized. And we see a similar sort of progress in the evolution of medical devices as well, where we're shifting from medical implants that were really sort of suitable for sort of a multitude of patients towards much more personalized, customized devices that are patient specific. And 3D printing has really enabled a lot of this personalization of medical devices. Here at Aspect, we think there is going to be a marrying of these sort of two fields to the point where we'll be 3D printing implantable cellular therapeutic devices. And so that's where our company is focused, and I'll tell you more about how this technology is being developed and how it may be regulated in the future. So in our discussions with Health Canada and other regulators, there's obviously some interesting challenges in trying to regulate advanced therapeutics that may be manufactured using non-traditional means. And so if you talk to Health Canada, they have kind of five main verticals, if you like, or five pillars of their innovation agenda. And these pillars include sort of pathways for clinical trials, traditional drugs and devices. But really what I want to focus on today is the pathway for advanced therapeutic products, or ATPs. And this is really a pathway that Health Canada and other regulators around the world are developing similar pathways, whereby it's a more flexible approach that tries to deal with these non-traditional advanced therapeutic products that are being developed using manufacturing processes such as 3D printing and bioprinting. So what is bioprinting? Well, really what we do is we take the technology of 3D printing, and 3D printing is where you design a structure using a computer software, and that structure is realized layer by layer to build up a three-dimensional structure. Now, in this case, we're using combinations of cells and biocompatible materials, such as alginate and collagen, to build living three-dimensional structures. These structures can be designed and built in various different sizes and shapes, but importantly, often these structures contain living cells. And so The printed structure has to be cultured over time to mature and develop. And what we see here is a microscope image of printed cells that over time are developing into interconnected networks. So these tissues are alive, but they're also importantly functional. And so this is just an example of a smooth muscle ring contracting and relaxing in response to appropriate therapeutics and contractile agonists. So we're able to develop and print tissues that are not just living tissues, but are also tissues that exhibit real function. Now, what we've done at Aspect is really leverage the technology of microfluidics in order to develop a unique approach to 3D printing. And in order to understand how this technology could be regulated, I think it's important to go into a little bit of detail as to how the technology works. And so effectively what we have is a 3D positioning system where the print head is a microfluidic device. What you see on this slide is just a schematic of one of our print heads. In this case, we have a print head that's able to switch between 4 different materials. These are represented by different colors on the slide. Those 4 materials could be different cell types or different biomaterials. Those biomaterials are loaded into the print head in a liquid phase, so it's a pre-cross-linked biomaterial, and then as the material is routed through the print head, it's brought into contact with a chemical cross-linking fluid. It's a non-toxic cross-linker, and that turns that liquid biomaterial into a solid cell-containing fiber almost instantaneously. And so it's that cell-loaded fiber, it's about the width of a human hair, that is deposited from the nozzle of that print head, and we build the tissue layer by layer using those cell-containing fibers. Why do we use microfluidics? Well, it enables us to do a few things that are unique to this technology. We can switch seamlessly between different cell types and different materials during printing. It's a very low shear stress environment, so we're able to print with fragile cells such as iPSCs and ESCs without triggering cell death, without triggering inappropriate cell differentiation. So it's a relatively cell-friendly approach to 3D printing. And it also enables us to combine multiple materials in the same fiber to generate what we call core shell fiber architectures. And so this is, again, a relatively unique function of this microfluidic approach to printing. So what do we do as a company? Well, we are trying to build what we call an end-to-end tissue solution. And so we have the 3D printer itself, the microfluidic print heads that I just described. We generate our own biomaterials. We have our own software that we use to design the tissues and drive the printing of the tissues. And then we partner with academics and commercial partners to develop and validate the tissues that we develop. And then the ultimate goal is to commercialize these tissues as clinical therapeutic solutions. And there's sort of regulatory input at almost every stage in this end-to-end process. I mentioned we had a couple of different types of partnerships, and again, I think it's important to understand how these work to understand how they could be regulated. And so we have a kind of a 3-pronged business model. We have partnerships with academic groups that make up what we call our discovery ecosystem. These are expert academics who have our printing system in their labs, and they use that system to generate and build tissues that they are already experts in. And as those tissues are developed, we are able to kind of help the development of those tissues and then partner with commercial partners to further develop and ultimately commercialize those tissues that are developed in that academic ecosystem. We also have central R&D within Aspect itself that is working away to develop specific tissues of interest as well, and I'll describe a couple of those in a second. So at Aspect, we have programs to develop preclinical tissues, so tissues that are for drug testing, tissues for toxicity screening, and disease modeling. And so there's 3 of those listed on this slide, we have a liver program in development. This is partnered with a Japanese firm to basically develop a preclinical model of drug-induced liver injury. We have a muscle tissue in development that's able to model contractile diseases such as diseases of the airway like asthma and COPD. We have a variety of other muscle tissue types in development as well. We also have a partnership on developing an immuno-oncology tissue. And so this is effectively printing microtumors using patient-derived cells that are really designed in a way to reproduce the microenvironment of the tumor. And so these can be used as a screen for novel immuno-oncology therapeutics. So those are the preclinical tissues that we have in development. But we also have core programs at Aspect where we're developing tissues for actual implantation into patients. So these are therapeutic tissues. We have a orthopaedic track where we're developing an implantable knee meniscus cartilage tissue. And we also have a pancreatic tissue in development, which is effectively a beta cell containing tissue patch that is an implantable for the control of blood glucose in patients with type 1 diabetes. What I'm going to do for the rest of the talk is focus really on one of those therapeutic programs and give you some idea as to the parameters that we control as we're developing and manufacturing these tissues, and then kind of move more towards the quality control and the regulatory pathways that these particular tissues could be exposed to. And so I'm going to start with the knee meniscus implant as the kind of example tissue that I'll take us through. And so the knee meniscus is one of the most commonly damaged areas of the knee joint. It's basically the shock absorber in the knee. You have two menisci in each knee, a medial and a lateral meniscus. And these are fairly commonly torn, particularly if you have an ACL injury. And there's around 1 million knee meniscus repair operations per year in the USA alone. One of the challenges with meniscus repair is that the tissue itself isn't really a pro-reparative tissue, and so it doesn't heal particularly well. And often the best short-term solution is a meniscectomy or a partial removal of the torn part of the meniscus. Unfortunately, in the long term, that leads to osteoarthritis in many patients. And so really there's a need to develop a different approach to meniscus repair that avoids that development of osteoarthritis in the long term. Synthetic meniscus replacements have been developed, but there aren't really any that have been adopted widely by the clinic, and there's various reasons for that. Mainly the lack of mechanical performance of those meniscal implants. So what we're doing at Aspect is developing a patient-specific bioprinted partial meniscus implant that would be sutured into place into the patient and replace that damaged area of the tissue. And that's really explained on this slide that demonstrates our vision for this particular bioprinted implant. So a patient would come into their orthopedic clinic, they would have an MRI scan of their injured and their healthy knee, and from that scan we would be able to generate a patient-specific design for their particular meniscus that's been injured. From that design, we would be able to print that customized implant that is just the right size and shape for the patient. Sizing is a particularly important factor when it comes to meniscal implants. If the meniscus implant is the wrong size, then it doesn't absorb the shock in the right way, and you still get damage to the articular cartilage. And so the accurate sizing of these implants is a very important factor when it comes to having an implant that actually functions effectively in the joint. So once the implant is printed, it would be inserted into the joint using fairly standard arthroscopic techniques. So this would be keyhole surgery the patient, the surgeon would suture the implant to the surrounding healthy meniscus tissue, and then the patient would enter their rehab and recovery pretty quickly after the surgery. So we're hoping that this would shorten the time to the patient getting back to pretty much full activity levels. So if we're 3D printing these implants, what sort of parameters are we able to control, and what parameters are important to regulating the function of these tissues? Well, I mentioned that we have software tools that we use to design and control the printing of the implant. And these software tools control things like the tool path that the fibers are laid down in as the tissue is printed. So each layer of that tissue can contain a orientation of fibers that are specific, and that can vary between layers. And so the orientation of these fibers are particularly important in a mechanically strong structure such as the meniscus, because the direction that those fibers are arranged in really dictates the mechanical characteristics of the final structure. So we have software control over the tool path, the infill density, so the density of the fibers, the distance between the fibers, and this can be varied on a layer by layer basis. And that has really important impact on the mechanical performance of the tissue. The biomaterials that we use in these tissues is also obviously an important parameter, in fact, a vitally important parameter. And one of the challenges with 3D printing, especially in the orthopedic space, is that the currently accepted materials for joint repair, such as ceramics, cobalt chromium, PEEK, etc., which are used in things like hip implants and total knee replacements, these aren't really suitable for 3D printing, and certainly not suitable for bioprinting. And so we're having to iterate and innovate when it comes to the types of biomaterials that we're able to use. And so, for example, in the implants that we're developing, we have a focus on a combination of synthetic materials such as polyvinyl alcohol and biological materials such as chitosan, both of which have been used in previously approved medical devices. But we have to guarantee that these materials that we're using are sterile, they're able to be sterilized, and also that they're biocompatible. And so we obviously have to perform in vivo and in vitro tests to ensure sterility and biocompatibility. One other component of the biomaterials is the cellular component. And so, in some of the implants that we're developing, we obviously want to include cells. This may improve the engraftment and repair of that implant after it's been sutured into place. And so obviously bioprinting enables us to include cells in the actual scaffolds during printing. And so rather than having to generate a structure and then seed cells on afterwards, with bioprinting, we're able to include cells during the process of printing, and so they're already embedded within the device. Obviously, there are regulatory challenges with cell-containing devices versus cell-free scaffolds, and often the materials that we use to print with cells are relatively mechanically weak. And so again, there has to be innovation on the cross-linking mechanisms and material types that we use when we're printing with cells. This also brings some challenges when it comes to sterilization. It becomes more challenging to sterilize an implant that contains cells versus a cell-free scaffold. And so the manufacturing of the implant with cells obviously has to be performed in sterile conditions, and that sterility has to be guaranteed. A final advantage when it comes to 3D printing devices is that the manufacturing of those devices can potentially be outsourced to distributed sites. And so what I mean by that is you could have multiple clinics all around the world who are printing these implantable devices. And so that point of care manufacture is something that's almost, when it comes to device manufacture, is almost uniquely suited to 3D printing. But distributed manufacturing also raises challenges when it comes to regulation. And so, for example, the question of who would be liable if a device that was printed in a clinic, if that device caused an adverse event in a patient, would the liability rest on the clinic that manufactured the device, or would it rest on the company that was providing the manufacturing technology to build the device? And so this distributed manufacturing kind of enables scaling, and it enables more personalization of the implants, but it does raise some important regulatory challenges versus the sort of more traditional centralized manufacturing strategy that we're kind of used to when it comes to more drug-based therapeutics. So I raised some of the parameters that are sort of under our control when it comes to 3D printing, but these parameters also need to be considered and recognized when it comes to kind of quality control and working to certain standards to develop consistent and safe implantable devices. And so what I've done here is I've kind of split these different considerations up into three main verticals. We have the raw materials that go into the medical devices. We have the biofabrication tools and technology, and we have some post-fabrication steps as well. And so if we focus first on the raw materials that we're using in these bioprinted implantables, obviously cell type and source is essential to consider. The manufacturing of those cells that we're using is a very important consideration, and having GMP manufacturing at the right time for those cells is essential. Other factors such as passage number of the cells, confluency, cell culture media, and culture protocols. These are all factors that really figure into the cell culture protocols and the quality and reliability of the cells that we use in these implants. Other material-specific considerations can include things like the specific sort of subtypes of biomaterial. So alginate is a material that we often use. It's a biological polymer, and so we have to really carefully maintain consistency of this biopolymer. And so things like the mg ratio in alginate is a particularly important factor when it comes to consistency of alginate. When we're working with materials like chitosan, the deacetylation percentage of chitosan needs to be consistent, and this is an important consideration when it comes to biocompatibility of this particular molecule. Molecular weight is obviously something that's important as well when it comes to biomaterials. For the biofabrication process itself, I mentioned that software designs and toolpaths for the fibers that we lay down have a really significant impact on the performance of the tissues themselves. And so having consistent control over the software that we use to design these implants, as well as the appropriate sort of recording mechanisms for ensuring that we know exactly what the design of each implant was, and if there are any variations in the software design, we need to record those variations. The processes involved during bioprinting, such as the concentrations of the biomaterials and the crosslinkers that we use, the flow rates of the materials going through the print head, and the actual print speed, these are all factors that really impact the size and quality of the fiber that's being deposited. And so again, these need to be recorded and controlled appropriately. There's also various sort of QA and QC metrics that need to be put into place when it comes to the printing platform as well as the print head itself. And I'm just going to go through a couple of examples of how the microfluidic printheads that we use for our printing processes are quality controlled. So this slide just kind of goes through at a fairly high level some of the steps that are involved in the quality control of the microfluidic printheads. First of all, we manufacture these printheads in a clean room environment. And then once we get these parts in, these are basically injection molded silicon parts. They're visually inspected to the first of all, there is no contamination in the channels of the printheads, and particularly sort of in the important regions of the printhead, like intersections and the valve regions. We need to make sure that there's no obvious defects in the printhead itself. The parts that pass this visual inspection are then used to manufacture the printheads themselves. And then those printheads are run through a series of automated tests. And these are tested for basically 5 known failure modes, various forms of fluid leaking from the channels, basically. We do cycle testing, what we call a burn-in, and this eliminates any devices that are going to fail early in life. We do some hydrodynamic characterization, and so this is basically where we measure the performance of the of fluids within the channels of the printhead, and so we make sure that fluids are running through the printhead under the appropriate pressures at the appropriate flow rates. We ensure that the valves that are on these chips are operating at the correct pressures, and then all of this data that we generate during the quality control of the printhead is stored on a microchip that's actually on the printhead. This is an RFID microchip that is able to communicate with the actual 3D printer when the printhead is loaded onto the machine. And so what that RFID chip does is it tells the printer exactly what kind of microfluidic printhead it is, so the printer can set itself up in the appropriate way. But it also stores all that QC data, and so if there is a failure in the field of that printhead, we can go back to that QC data and try to analyze what the failure mode was and potentially alter our QC metrics to ensure that that same failure doesn't occur again. So there's really robust testing and quality control of these microfluidic printheads because these are effectively the core of that manufacturing process to generate these 3D printed tissues. And then post-fabrication, there are various quality control measures we have to take as well. And so some of the implants that we generate need to be post-printing. There are processing steps to either cure or culture the implants. Biomechanical and biofunctional tests are essential. And for the meniscus implants that I was describing earlier, these can include non-destructive tests such as unconfined compression, compressive modulus type tests. We can also take one representative implant from a batch and perform destructive tests such as suture retention strength. And then for the other cell-containing implants that we generate, biofunctional tests can include things like insulin release from the pancreatic tissue patch that we're developing, or immunoprotection of that pancreatic tissue patch. So the QC measures of each tissue type is really customized to the specific tissue that we're developing. So the manufacturing process, the biomaterial composition, the cellular composition, and the post-fabrication quality control measures are really all customized to the specific type of implant and device that we're developing. And so really when we have this kind of unique collection of parameters that are involved in the manufacturing and validation of these bioprinted implantables, and each type of implantable has different composition and manufacturing processes. How can they possibly be regulated under a single pathway? Well, the answer basically is that they can't. And Health Canada have kind of realized the same thing. And so what they've done is they've created this new pathway for advanced therapeutic products. And really, this is intended for devices or drugs or combinations of devices and drugs that are so novel and complex or distinct that they don't really fit within the existing regulations. I previously showed a slide of the 5 pillars of Health Canada's innovation agenda, and this slide shows a more detailed visual of that advanced therapeutics pathway, or advanced therapeutic products pathway, or the ATP pathway. And so if I can summarize what this ATP pathway really is, what they plan to do at Health Canada is continue to regulate kind of most drugs and devices under an existing framework. And so that's on the left-hand side of this slide. Most drugs and products would still be regulated through that existing drug, medical device, cell, tissue, organs, or blood kind of framework. But if something doesn't fit into that framework, then this ATP pathway offers the ability to create a more tailored requirements and design for a regulatory framework, what they call a sandbox, specific to that product area. And so what would happen is the tailored requirements are intended to be created through this kind of iterative consultation with stakeholders and subject matters experts. And then that specific tailored requirement can be added to the Food and Drugs Act through a ministerial order. I think technically they added to something called a Schedule G of the Act. And this means that no additional regulations are required. And so from that point on, you would have this kind of custom regulatory process that would be applied to that particular device, that particular implantable device. And then once this sandbox is created, the sponsors of that clinical trial can then submit a license application for market authorization. And so, I mean, this is in development, and other sort of regulators around the world are, I think, thinking along similar lines and trying to put similar structures in place to deal with these unique medical devices and products. And I mean, Health Canada are kind of in the process of building this, and my understanding is they're conducting sort of further engagement sessions on this ATP pathway as they work through the implementation process. They also have what's called a concierge service, which is intended to kind of help sponsors through this whole sandbox process and really kind of hold our hands as we go through this relatively new process of getting these customized regulations in place. I don't know if that's in place yet, but I think it's going to be probably summer or fall this year that concierge process would be activated. And so with that, I'd like to say thank you very much, and obviously I'd love to take any questions that you might have.
Great, thanks so much for that presentation. Very interesting. I especially like seeing sort of the new process that Health Canada is going to be going through laid out nicely like that. So once again, if you have questions, you can type them into the chat. If you feel like asking your questions out loud, you can unmute yourself by clicking the little microphone and asking. But I'm just going to get started with some questions from Hassan and Sari. So Sam, I think I heard the print heads, are they injection molded? Is that the fabrication type for the chips?
It is, yeah. So the print heads that we generate at scale. So when we send print heads out to many of our kind of academic users around the world, those tend to be generated at a larger scale, and so injection molding is one approach we use to achieve that scale.
Great. And are there different types of print heads to get? For example, I think you mentioned the core shell structure. Are there any that are 2 layers or 3 layers? are they generally just one type of print head for all of your different prints?
No, that's a very good question. And we make multiple different types of print heads. And so some are able to switch between 2 materials, some between 4 materials. Others are able to generate those core shell structures that I showed on the slide. And so absolutely, kind of each print head is kind of customized to do to generate a specific type of fiber or handle specific types of biomaterials. And so it's kind of a way of increasing the flexibility of the printing system whilst maintaining consistency of the underlying platform, because each of these print heads just slots onto the same 3D printing platform.
Great. So we have a question from Jeff Lomax. Do you want to unmute yourself, Jeff?
Yeah, I think I'm unmuted. Can you hear me?
Yep, hear you loud and
Yes.
clear.
Terrific. Great presentation. Thanks a lot. It's the example you give is terrific in the sense that, this is a space where I think when we started the talk, it was framed around, wanting to get the best science for patients, but wary of sort of charlatans. And certainly knee and cartilage is an area where we have a lot of questionable work going on. I just wonder if you could comment at all in terms of, the approach that's going on with this device in terms of kind of the evidence that's guiding it, any sort of preclinical, just the kind of weight of evidence around why this is an important approach from a standpoint of, clinically for knee replacement, particularly in light of the fact that there's a lot of other things going on that are, probably not subject to the same rigor.
Yeah. No, thanks for the question, Jeff. I think it's a really good one, and I think it doesn't just apply to orthopedics, but I think there's quite a lot of technologies out there that to some extent overstretch their promises, and so bioprinting in general, I think, is it's an easy technology to kind of envisage what the potential use of that technology could be. So you've probably seen sort of promises made about we'll be able to print whole organs in the next few years and things like that, and I don't think that's really how the field should be positioning itself. I think we need to start with implants and tissues where we think 3D printing and bioprinting can offer an immediate advantage to or solve an immediate clinical need. And in the orthopedics field, this approach to generate more personalized meniscal implants, I think, is one where through consultation with orthopedic experts and surgeons and orthopedic companies, we've realized that there is a real unmet need there, and when we've explained our approach to them, they see genuine potential there. And so we really took a lot of input and advice on this program. But during the development of the technology itself, the development of the meniscus technology, we obviously have to follow the science, and we perform in vitro studies to maximize mechanical strength, and but we also have in vivo studies in place where we investigate things like biocompatibility of the implants. We have animal models where we're investigating the safety and efficacy of these implants in a more physiologically relevant environment. And so our goal is to develop a data package for these implants that we can take to the regulators, and they will effectively enable us to enter clinical trials. And the only way we can do that is to have real scientific rigor in the development process and do the right experiments to demonstrate safety and efficacy. I don't think if you're going to go to the regulators and persuade them to allow you to go into the clinic, there's no other way around it, really.
So we have a couple questions coming in from the chat on whether or not the print heads are disposable or reusable, or how you would sterilize those between uses, and also how much they cost. Could you address that?
Yeah, sure. I mean, um, the cost is. I mean, these are, I can't tell you exactly how much they cost,
Sure.
but you know, they're a few $100s. They're not tens of thousands of dollars.
Okay.
Um, and they are disposable devices. So, what we would recommend is that you would basically do one print session. So you may print a batch of tissues, and then the next day when you came back to the lab, you would slot in a new print head and then print the next batch of tissues. So, in order to avoid things like cross-contamination or between different tissue types, or even just contamination of a structure, you would use a fresh sterile print head for each print.
Sure. A question from Craig, who says, you mentioned bioprinting the cells to preserve viability, but are you able to print aggregates or clustered cells and maintain the aggregates intact?
Yeah, again, good question. When we first started printing with cells, we were printing sort of suspensions of cells, and so they were, effectively single cell suspensions, but at pretty high density. We can go up to several hundreds of millions of cells per milliliter, which is a very high dense structure. But with our programs like the pancreatic program, we had to modify our printing technology to handle clusters of cells. And so we do print with primary pancreatic islets as well as clusters of stem cell derived cells, and some cell lines we print as clusters as well now. So yeah, more recently we have been printing with clusters and aggregates of cells.
Very interesting.
And I should say as well, the process of printing doesn't break up those aggregates, so they maintain their sort of aggregate structure as they're printed.
Yeah, I can imagine important for different applications. So Adrienne would like to know, what are the current standards in terms of cell biology? What baseline do you need to produce these devices? For example, cell viability, cellular function. And do you decide the guidelines yourself, or are there external guidelines that you're following?
Um, so I should sort of qualify that the answer with we haven't gone through the process of taking a cellular device through the approval process yet. So I
Okay.
don't know the answer to that 100%. But
Yeah.
what I can say is that the parameters that he laid out are definitely involved. So ensuring that post printing the cells that you've used are viable, and that could well be not just immediately post printing, but several days post printing or even longer, depending on what the lifetime of the device is supposed to be in the patient. And then
Right.
when we look at function of those cells, I mean the function really depends on the type of implant. And so for something like the pancreatic patch device, the function would be more focused on insulin release in response to glucose. And I mean, there you would probably have in vitro QC measures in place where you're basically exposing the printed implants to different concentrations of glucose and confirming that you have the appropriate insulin release. So, in addition to viability, there would definitely be sort of function specific endpoints that you'd have to measure as well. And, those would be not just in order to kind of persuade the regulators that something was efficacious and safe, but also, as a fairly standard QC measure, once these devices are being used in the clinic, you'd also have to sort of continually measure those kinds of endpoints as you're manufacturing these devices.
Okay. Just a second, these questions are coming fast.
That's good.
It is. Lots of interest here. So Marco wants to know if you can print vascularized tissues.
Yeah, I mean, we can certainly print with endothelial cells, and so we have work going on where, we're printing with HUVECs and other types of endothelial cells.
Right.
With the core shell printing approach, one way we can leverage that is where the core of the fiber is not a solid material, but it's actually a liquid that just basically washes away during printing. And so what you end up with is a hollow fiber with a hydrogel shell, but a hollow core. And the size of those fibers can be modified just by changing the pressure settings on the printer. And so you can generate fibers that have a relatively narrow core of just tens of microns in diameter, or you can generate fibers that are much larger diameter with a wider core. And so you can kind of model some different size vessel types by
Yeah.
using that hollow core shell printing approach.
I think that addresses one of the questions I may have skipped as well. So you can control the pressures of the different channels independently from each other, correct?
That's correct, yeah. Yeah. So each of the different channels on that printhead, regardless of the type of printhead, has a different pressure input. And so you can control the flow rates independently in those different channels, and basically the relative flow rates in the channels dictate parameters like the diameter of the fiber that's generated, and obviously the speed of printing as well.
Right. So do you have a list of the different types of available print heads, or would you be able to work with different people to develop new print heads?
Yeah, we like our website has sort of a list of some of the printheads that are available, but the way we work with most of our partners is we understand kind of what their research is, and based on that and through conversations with them, if it's obvious that they really need a custom printhead that performs a specific function, then we sort of talk to other people about whether that would be useful to them as well. And if we kind of think actually this could be a good design for a printhead or a good use for a printhead, then
Yeah.
we'll design and build it and then offer it broadly to the group.
So we have an anonymous question asking about if you're working on producing kidney organoids via bioprinting.
We are not focusing on kidney yet within the company itself, so none of our internal programs as yet are kidney focused, but we do have kind of academic partners who have kidney projects, so printing with sort of kidney cells. I don't know whether they're printing with kidney organoids or not.
Okay.
I'd be interested in talking to whoever it is, though, that is thinking about doing it.
All right, you hear it, guest. You can get in contact with Sam and ask your questions there. Lisa Caddick also has a question about the immune response to your meniscal product or any of your other products. Have you looked into that at this stage in your work?
Yeah, that's a good question, Lisa. We have, and there's definitely an important consideration, and so some of the materials we use have been used in non-printed implants, over the last couple of decades, and so the kind of host response to some of the materials that we're using is pretty well known, but we still have to confirm using sort of fairly standard in vivo models whether or not we have an inflammatory tissue response to those materials. And if we do, then we have to modulate or modify the specific type or subtype of material that we're using to kind of dial down that immune response. So yeah, it's a good question, and we definitely take that into consideration when we're developing these materials and implants.
All right. Sam, would you have a couple minutes if we run a little bit over time to stay with us and answer a few more of these questions?
Yeah, sure.
Great, thank you. So Kabilan says mostly your bioprinter employs chemical cross-linking methods. Do you have a plan to use photo cross-linkable materials? Mostly, he's looking specifically at gelatin since it's more cell adhesive than alginate.
Yeah. Um, Again, yeah, very good point. And so uh, he's absolutely right. So alginate is kind of almost biologically inert, in the sense that cells don't tend to interact with it, which is good in some ways, but challenging in others. And so we also load extracellular matrix factors into the alginate material as we print. So we make what we call a bioink, and that bioink contains alginate and other biological factors like collagen. And because the bioink contains collagen, the cells are able to interact with it, and so they're able to kind of spread and form networks within the printed structure, even though the alginate is there and it's relatively inert. So that's one way to sort of get around that problem, or that challenge. Using photo cross-linkable materials like Gelma and Pegda, it's definitely possible with this system. Again, you can load those materials into the alginate to form a cross-linking step post-printing, and then you can even remove the alginate if you don't want it to hang around using non-toxic enzymatic methods. We are looking at ways of performing photo cross-linking directly on the print head during printing, and so using that as a cross-linking approach instead of the chemical cross-linking approach. So it's definitely something we're looking at, yeah. So I would say keep watching this space, and hopefully we'll have something on that to show you fairly soon.
Yeah, very interesting to hear about the different work going on with the biomaterials that you're using. So we also have a question that you have some job postings up online. Would you have any recommendations on strategies that people can use, or what specifically you're looking for in these job postings?
I approve of whoever's asking that question. So my advice is, I mean, there are several different job postings on the website, and each different role has quite different requirements, and so I would say that in general we're open to just interviewing people that have relevant experience, and that experience doesn't have to be within industry, it doesn't necessarily have to be within bioprinting, but we're looking for people who are kind of experts in a relevant field. And I would encourage you to take a look at our website, look at the job descriptions, and if you think you're a good fit, then please get in touch and send us your resume, and we'll definitely consider you.
So shout out to, that was Rachel Gerlin who asked that question in terms of just general advice. Apologies for mangling your question a little bit. So there's another question from Craig who's asking, are there any manufacturing concerns or strategies around printing in a BSC? Or yeah, so could you address just how you're keeping your product sterile when you're printing it?
Yeah, the printing system that we developed is sized so it will fit inside a BSC.
Okay,
Now, it won't fit inside every BSC because BSCs tend to come in different shapes and sizes.
for sure.
So there are certain recommended ones that we use for our own work, and we recommend that our partners use for their work as well. But yeah, I mean, I would say that if you're doing sort of cell-free scaffold printing or biomaterial development, then printing on the bench is okay. But as soon as you're doing cell printing with sensitive cells, primary cells, anything like that, then we'd recommend you using it inside a BSC.
Okay.
The print heads themselves and the sort of all of the tubing that basically leads from the cell and material reservoirs into the print head, they're sterile. So all of the component parts that come into contact with the cells and the biomaterial are provided sterile.
Good to know. Have you considered, Sarah Mishriki asks, have you considered the use of prefabricated 3D cellular structures like spheroids as opposed to single cell suspensions? I think you mentioned this a bit, that you were able to print aggregates. Are you still looking at that?
Yeah, I think it's a very similar question to that previous one. But yeah, the answer is yes. And so there's with certain cell types, there's lots of evidence that growing them as an aggregate improves their differentiation, or is actually an essential part of the differentiation process. And so for projects like the pancreas project that we have, printing with clusters of islet-like cells is potentially an important part of that tissue manufacturing process. And there's probably many other cell types as well where printing with an aggregate may improve the performance of the tissue versus a single cell suspension. I would say though that one of the sort of advantages of using a system that's able to print with biomaterials that are very low viscosity at low concentration. So we're able to print with alginate at less than 1% concentration. So it's a very low viscosity material. And so the reason we can do that is because the alginate is cross-linked on the chip. And so, the fiber that's pre-solidified is deposited out of the nozzle. And so when you have such a low concentration of the alginate biomaterial, you can actually kind of ramp up the concentration of biological factors like the collagen I mentioned, and you can also increase the density of the cells as well. So, even if you aren't printing with aggregates, we can also print with very high concentrations of cells and still maintain the ability to pattern fibers in 3D. So I think there's we're closing. If you don't print with aggregates, we're pretty close to almost printing with like a solid cell fiber anyway.
Wow, that's very interesting and pretty impressive. So one last question from Vanessa is, what would you say the biggest hurdle is in terms of recruitment and onboarding of employees? Is there anything missing in the training of the people that you hire?
Ooh, what a good question. I think, it depends on the position. We have
Sure.
on the R&D side, we have cell biologists, we have biomaterial scientists, we have on the engineering side microfluidics experts, we have software designers, we have a variety of different skill sets, all contributing towards the development of the printing technology as well as the tissues that we're developing too. And then obviously we have the business side where we recruit people on the for various other parts of the business too. So it all depends on the specific role that we're recruiting somebody for as to what we want their kind of training, their pre-aspect training to be. I would say though that we always recruit people with, certainly on the R&D side, with a good solid scientific or engineering training background. So usually at least a master's, if not a PhD. But problem solving is something that often isn't part of the training for scientists, but is a really important skill set to have especially in a a relatively small company where you may be faced with scientific challenges or technology challenges that require some out of the box thinking, and so that's a sort of a soft skill that isn't necessarily obvious on paper, but we do try to sort of assess during the recruitment process, and certainly early on in the once we've hired somebody, we do need to make sure that they're able to do that kind of out of the box problem solving.
Yeah, so the soft skills are important and very hard to teach, I can imagine. Okay, so I think that is all of the time that we have. Thank you, Sam, very much for teaching us a bit more about your 3D bioprinter. If anyone has questions, the link to Aspect Biosystems is here on the page. Thank you everyone so much for joining us, and tune in next month for another webinar.
Yeah, thanks everybody, and stay safe.